You've probably heard of the low-carb, high-fat diets such as the Atkins diet, and in this lecture, we'll see just how much ATP is made from fatty acyl chains as well as the limitations to using fat for energy. In this lecture, we're going to see how fat molecules are broken down and transported to the cells that need them. We've already talked about the role of mitochondria for ATP synthesis, and the breakdown of fatty acid chains and beta oxidation will also occur in the mitochondrial matrix. You'll see that we'll also be producing those reduced coenzymes, NADH and FADH2, and we'll compare how much energy can be made from fat versus glucose. There's only one section of Stryer for this lecture if you want to take a look at the supplemental readings. We've already seen in the lipids lecture that triacylglycerol molecules are the energy storage form of lipids. Fatty acid chains are linked to a glycerol backbone via ester linkages, and these can be found in fat cells, which are also known as adipose tissue, but you can also find these molecules in muscle or other tissues like the liver. The chains are usually found in the saturated form since this allows for the tight packing, and when it comes to fat tissue, there pretty much is an unlimited amount you can store in this format. In order to catabolize these chains, we do need to release them, and this will be through the hydrolysis of the ester bonds. It's the GPCR signaling pathway that we've seen before that can lead to the breakdown of triglyceride molecules. In order for the lipases to be activated, they do need to be phosphorylated, and this is all going to be mediated by protein kinase A, which we saw was activated by cyclic AMP produced by adenyl cyclase, and adenyl cyclase was activated by the GTP-bound G protein. The lipase that's activated is sometimes known by the name of hormone-sensitive lipase, so this might be an enzyme you've heard about before in your physiology courses, and it's going to use a water to release free fatty acids from the glycerol backbone. There is a monoacylglycerol lipase that will remove that last fatty acid tail, producing the soluble glycerol backbone as well as the fatty acid tail. Remember, fatty acids are amphipathic in nature, and they do require a protein transporter called albumin to transport them to the cells that need to use them for energy. For instance, your muscle cells when exercising. And albumin is one of those proteins that might get tested for if you ever go in for blood work. It's able to transport a variety of hydrophobic molecules throughout the body in the bloodstream. Whenever we need to make energy, it's the catabolic hormones that are going to signal for the breakdown of triacylglycerol molecules via GPCR signaling. Epinephrine was that fight or flight hormone that is released from your adrenal glands, so when you're exercising, you're initiating the signaling pathways to help with fat catabolism. Under low glucose states, glucagon can also help with the generation of energy from fats through this GPCR signaling pathway, where we have GTP-bound G-alpha subunit being released, activating adenyl cyclase to make cyclic AMP, which then activates protein kinase A to phosphorylate and activate the lipase. Triacylglycerol molecules are very hydrophobic, so we need to first hydrolyze off the free fatty acid chains, which can then be transported to the cells that need them via albumin. This slide just summarizes that process. Triacylglycerol molecules are broken down from fat cells into the glycerol backbone and free fatty acids. And we've already seen how glycerol can actually be used by the liver cells to produce glucose. So you can't make glucose from acyl chains or acetyl-CoA, but we can make it from the glycerol backbone. The free fatty acids will then be transported by albumin to other cells, where they'll be broken down in beta oxidation, which is also known as fatty acid oxidation, to produce the reduced coenzymes NADH and FADH2. And then we're going to make a lot more acetyl-CoA, which can then enter into the citric acid cycle and generate even more NADH and FADH2. Now, remember these processes occur in the mitochondrial matrix, so we have to first look at what happens when a fatty acid chain arrives at a cell. When a fatty acid chain, also known as an acyl chain, where R indicates some length of a carbon chain, when it arrives at a cell, it needs to first be trapped inside the cytoplasm, and this involves the covalent linkage to coenzyme A. Remember, fatty acids are amphipathic in nature, and they could just sit in the membrane. So if we want to use them for energy or somewhere else in the cell, we need to first activate them using coenzyme A. The enzyme that catalyzes this reaction is called acyl-CoA synthetase, and it's going to use ATP as the energy molecule, but we actually make an AMP product where we couple it to acyl-CoA synthesis. The reaction that acyl-CoA synthetase is catalyzing is actually reversible under cellular conditions. It's the production of the pyrophosphate that's going to help drive this reaction in the forward direction. When pyrophosphate is hydrolyzed to two inorganic phosphates, this depletes the PPI product in a favorable reaction. So when PPI is hydrolyzed to two PIs, this will release 34 kilojoules per mole of energy because the two inorganic phosphates are significantly more stable. So we've activated the fatty acyl chains at the expense of ATP to AMP and the breakdown of pyrophosphates to two PIs. But there's a problem when AMP is made. The problem lies in the regeneration of ATP. Remember, when it comes to making ATP, the majority of ATP synthesis comes down to ATP synthase that uses ADP as the substrate. It can't use AMP as the substrate, so we need to sacrifice a second ATP molecule to make ADP from AMP. Adenyl kinase is going to move a phosphate from that second ATP onto AMP so that we now have two ADP molecules that ATP synthase can use. So whenever you see a reaction generating AMP, it's actually using two ATP molecules, one for the reaction and the second one to make ADP. So even though the breakdown of ATP to AMP plus pyrophosphate releases more energy than ATP to ADP, we don't see this reaction very often because of this second ATP requiring step. Okay, so we've made an acyl-CoA molecule in the cytoplasm, but it needs to get into the matrix where beta oxidation occurs. Remember, the outer mitochondrial membrane does allow for molecules to enter via porins, so that acyl-CoA molecule and ADP and PI can cross, but the inner mitochondrial membrane is impermeable to many molecules, so we do need a transporter to get the acyl chain in. And coenzyme A is actually a very big molecule. This is the structure, which I haven't shown you before, but it does have a lot of charged and polar functional groups on it, so it can't cross the inner mitochondrial membrane. The thiol group down here is where that acyl chain will be attached, activating it for the different pathways. And this is the same group that we saw on acetyl-CoA, where acetyl just means two carbons. Acyl means anything longer than that. Acyl-CoA is made in the cytoplasm, activating it for either energy production or even the synthesis of phospholipids. But energy production from its breakdown in beta oxidation takes place in the matrix of the mitochondria. The acyl-CoA molecules can pass through the outer membrane via protein porins, but can't cross the inner mitochondrial membrane since coenzyme A is quite large and water-soluble. Other than acyl-CoAs being used for phospholipid synthesis, can you think of a reason why it's made outside of the mitochondria if beta oxidation occurs inside the matrix? The answer is actually regulation. The import of fatty acid chains is regulated to ensure that they are only broken down when you need energy. Acyl chains are imported across the mitochondrial inner membrane by a carnitine shuttle. This shuttle involves a transferase called CAT1 that transfers the acyl chain from coenzyme A onto carnitine to form acyl carnitine in the intermembrane space. There's then a translocase that will exchange the acyl carnitine for free carnitine from the matrix side. Carnitine acyl transferase 2, or CAT2, will then transfer the acyl chain back onto coenzyme A using a free coenzyme A found in the matrix. So there are actually two pools of coenzyme A, some in the cytoplasm and some in the mitochondrial matrix, but they're separate pools since CoA can't cross the membrane. The acyl-CoA molecule can then be catabolized in beta oxidation to generate acetyl-CoA, NADH, and FADH2. Now that we've gotten our acyl chains into the mitochondria, fatty acid oxidation is going to generate acetyl-CoA that can be used in the citric acid cycle, as well as NADH and FADH2 that can be used in oxidative phosphorylation to make way more ATP. So let's start by looking at beta oxidation. Beta oxidation involves multiple rounds of 4 reactions. Each of these steps represents one round or 4 enzyme catalyzed reactions. Let's start with a fatty acid chain that has 16 carbons in the saturated form, and this will be attached covalently to coenzyme A. There are 4 steps that are going to require a water molecule and a second CoA. First, to oxidize the beta carbon and allow for the thiol cleavage to release an acetyl-CoA. Since there will be redox reactions involved, one NADH and one FADH2 will be made in each round of beta oxidation. Acetyl-CoA, which is going to be the other product, carries 2 carbons for catabolism in the citric acid cycle, and that will produce a chain that is now 2 carbons shorter, or 14,0-CoA, which can then go through another round of beta oxidation. So this cycle will be repeated, and you're going to get another NADH and FADH2, as well as another acetyl-CoA. The cycle will continue one more time. And then you'll keep repeating this cycle until we get to the very end. The last round that's going to start with a 4,0-CoA will actually make 2 acetyl-CoAs in the last round, along with one NADH and one FADH2. I'm not going to show you all of the reactions. You can look them up, but I do want to talk a bit about why it's called beta oxidation. If we start with a saturated or reduced chain of carbons, it's not going to be very reactive and it will be hard to break down. The 4 separate reactions are going to modify this beta carbon, where the first redox reaction will introduce a double bond between the alpha and beta carbon, producing FADH2. Water will then be added across the double bond, and then a second redox reaction will make NADH and create a keto group on the beta carbon. This will make it even more reactive. And remember, we saw how reactive carbonyls were in the carbohydrates lecture. So this will allow the thiol on coenzyme A to come along and bind to the beta carbon, releasing acetyl-CoA, which will include the original coenzyme A and the 2 carbons, including the carbonyl and the alpha carbon. The fatty acyl-CoA molecule is now going to be 2 carbons shorter and include the beta carbon bound to the new CoA molecule. These 4 reactions can then be repeated in the next round of beta oxidation. If we look at the overall reaction for beta oxidation, when you start with a 16 carbon chain, you're going to make 8 acetyl-CoAs. Remember, these all carry 2 carbons, so 16 divided by 2 is 8, but there's actually one less round of beta oxidation because the last round generates 2 acetyl-CoAs. Each round of beta oxidation will produce an NADH and an FADH2, so we'll have 7 of each of these. And then we're also going to require a water molecule and a coenzyme A, so 7 of each of those. Let's now walk through how much ATP is going to be made. Let's walk through how many ATPs are going to get made from that 16 carbon chain. So we saw that beta oxidation is going to generate 8 acetyl-CoA molecules, 7 NADHs, and 7 FADH2s, but it's also going to require 7 water molecules. Those 8 acetyl-CoAs will then enter into the citric acid cycle. So pause the lecture now and see if you can work out how many molecules are made from this point on. The citric acid cycle is going to run 8 times. So we're going to make 8 GTPs, 24 NADHs, so 8 times 3, and 8 FADH2s, but we're also going to require 16 water molecules because each cycle requires 2 water molecules. The NADHs and FADH2s from beta oxidation and the citric acid cycle will then pass their electrons to the electron transport chain. So we'll have a total of 31 NADHs and 15 FADH2s. When they pass their pair of electrons through the chain, remember complex 4 will make a water molecule. So we'll actually make 46 water molecules in ETC, or 31 plus 15. We can then use the PO ratios to figure out how much ATP is made. So 31 times 2.5 equals 77.5, and 15 times 1.5 will give you 22.5. If we add up these numbers, we'll get a total of 100 ATPs made by ATP synthase, along with 100 water molecules made at that beta subunit. Our grand total will then be 108 ATPs made, including the GTPs made by the citric acid cycle, and 123 water molecules being made, and all of these steps are going to be found in the mitochondrial matrix. If we completely oxidize a fatty acid chain that has 16 carbons in the mitochondrial matrix, we're going to require 108 ADPs and inorganic phosphates to make 108 ATPs. And we're also going to need 23 oxygen molecules, and remember this is half the number of NADHs and FADH2s that pass their electron through to complex 4 in the electron transport chain. Those 16 carbons will also make 16 carbon dioxides in the citric acid cycle, and will also make 123 water molecules as waste products. For the net reaction, only one coenzyme A will be released, but there will be multiple reactions where CoA is added or removed. So for a single chain, we're making 108 ATPs, and if you think about a triacylglycerol molecule, there's actually 3 fatty acid chains. So we'd be making over 300 ATPs, or 10 times more than a single glucose molecule. Now, if we do talk about that free fatty acid chain that came from adipose tissue, so the chain without the coenzyme A on it or attached, we would actually have to use 2 of those ATPs for the activation step and regeneration of ADP from AMP. We'll also need 1 water molecule for the hydrolysis of that pyrophosphate, so it'll be a total of 106 ATPs and 122 water molecules. So just to summarize, fat breakdown is dependent on the hormone signaling pathways. So this is the link between exercising or low-carb diets, leading to the enzymatic breakdown of triglyceride molecules. The free fatty acids will then be transported to the tissues for catabolism, and this does require albumin to transport these amphipathic molecules. Once they get into the cell, they do need to be activated through the addition of coenzyme A by acyl-CoA synthetase, and this does require 2 ATPs, one for the reaction and another to regenerate ADP from AMP. The carnitine shuttle will then import the acyl chain by first transferring it onto carnitine, and then translocate it in exchange for a free carnitine. The acyl chain will then be transferred back onto coenzyme A in the mitochondrial matrix, and this import step will be inhibited by the production of a molecule called malonyl-CoA, and that's going to be made in fatty acid synthesis. Beta oxidation will then break down the acyl chains in a series of 4 reactions, which will produce NADH, FADH2, and acetyl-CoA for the citric acid cycle. Now, not only do we need oxygen in oxidative phosphorylation to make ATP from fat, but there's also one other limiting factor when it comes to getting all that energy out of a fatty acid chain. When it comes to making all those NADHs and FADH2s in the citric acid cycle from the acetyl-CoA molecules, remember that the citric acid cycle is a cycle and is dependent on the amount of oxaloacetate that's present. So say we had hundreds of acetyl-CoAs coming in from beta oxidation, but we only had 10 oxaloacetates. That oxaloacetate is going to be the limiting factor, and you won't be able to efficiently make ATP from fats. We can speed up the citric acid cycle by producing more oxaloacetate, and there's a reaction that involves the use of pyruvate, which comes from glucose or glycogen. We did see this reaction when we were talking about the mutations that can be found in the citric acid cycle enzymes. So when it comes to efficiently producing ATP, it's actually better to have a blend of fuels, both carbohydrates and fats, in order to make a lot of ATP. When you don't have a blend of fuels, a phenomenon known as hitting the wall occurs, and there's a great representation of it in an old movie starring Simon Pegg, who's attempting to run a marathon without much training and figuratively and literally hits a wall. So if you're looking for something new to watch in isolation, you can find it through the U of T library. They have a bunch of movies you can watch. Hitting the wall happens when you've depleted all your glycogen reserves and can no longer supplement the citric acid cycle with the production of oxaloacetate from glucose to generate ATP from fat. So just in summary, in order to generate energy from fat, we do need to initiate the GPCR signaling pathways leading to the phosphorylation of the lipases that release the fatty acyl chains from the triglyceride molecules. These fatty acid chains can then travel through the body with the help of albumin in the bloodstream, and they'll then be activated in the cytoplasm by acyl-CoA synthetase, which requires 2 ATP molecules. The carnitine shuttle then controls whether or not the acyl chains are imported into the mitochondrial matrix, and this will use 2 acyl transferases and a translocase. And then beta oxidation, along with the citric acid cycle and oxidative phosphorylation, can generate a ton of ATP, provided we do have a blend of carbohydrates and fatty acids being broken down, as well as the availability of oxygen for the electron transport chain. I just want to leave you with a thinker question. Do you think taking carnitine supplements on their own could help you lose weight? Now, they were marketed as a magic pill years ago, but knowing what you know about biochemistry, how would you rationalize whether or not this would be a good idea? Check out the 2 brain break questions posted on Quirkus before moving on.